Deoxyribonucleic acid stores genetic information within a double helix stabilized by hydrogen bonds and a robust phosphodiester backbone. Breaking this structure requires overcoming a high kinetic barrier through enzymatic catalysis. The Cas9 protein is a large bilobed endonuclease derived from Streptococcus pyogenes. It consists of a recognition lobe for binding nucleic acids and a nuclease lobe containing the catalytic machinery. Cas9 remains inactive until it binds a single guide RNA molecule. This synthetic fusion of CRISPR RNA and transactivating CRISPR RNA creates a functional ribonucleoprotein complex ready for genomic surveillance. Once loaded, the complex scans the genome by diffusing along DNA strands. It utilizes 3D collisions and 1D sliding to locate specific target sites amidst billions of non-target base pairs. The search is governed by the protospacer adjacent motif, typically a 5' NGG sequence. Cas9 only attempts to unwind DNA if this specific chemical signature is detected.
Within the PAM interaction domain, two specific arginine residues form hydrogen bonds with the guanine basis of the PAM. This interaction acts as a molecular key to initiate DNA melting. Following PAM binding, the Cas9 complex forces the DNA helix to unwind locally. This exposes the target strand, allowing the guide RNA to test for complementary base pairing. As the guide RNA binds to the target strand, an R-loop structure forms. The non-target DNA strand is displaced and stabilized by the protein, while the RNA-DNA heteroduplex expands. The first 10 nucleotides adjacent to the PAM constitute the seed region. Perfect complementarity here is essential for the complex to proceed toward a stable conformational state. If the seed region matches, the heteroduplex continues to form across the full 20 nucleotides. Mismatches in the distal region can lead to premature dissociation, preventing off-target cleavage.
Full complementarity triggers a major rearrangement of the recognition lobes. This mechanical movement acts as a checkpoint, ensuring that the nuclease domains are only activated at the correct site. The H&H nuclease domain undergoes a large rotation to position its catalytic center near the target strand. This movement is the final requirement for the chemical cleavage step. Simultaneously, the RUV-C domain aligns with the displaced non-target strand. Cas9 uses these two distinct nuclease sites to ensure both strands of the DNA helix are severed. The H&H domain utilizes histidine and asparagine residues to coordinate magnesium ions. These ions polarize water molecules to initiate a nucleophilic attack on the phosphorus atom. Two magnesium ions are coordinated in the active site. One activates the nucleophile, while the other stabilizes the transition state and the leaving group of the broken phosphate bond.
The H&H domain cleaves the target strand exactly 3 base pairs upstream of the PAM. The covalent bond is broken, leaving a 3' hydroxyl group and a 5' phosphate. The RUV-C domain performs a similar catalytic reaction on the non-target strand. While the H&H domain is highly flexible, the RUV-C domain remains relatively static within the nuclease lobe. The result is a double-strand break with blunt ends. This physical disruption of the DNA helix is the signal that triggers the cellular DNA repair machinery. After cleavage, Cas9 remains tightly bound to the DNA ends for an extended period. This high affinity prevents the broken ends from immediately reannealing before repair proteins arrive. Eventually, the complex must dissociate to allow repair. This can be facilitated by mechanical forces or the movement of ribonucleic acid polymerases along the DNA strand.
The cell often repairs the break using non-homologous end joining. This process frequently introduces small insertions or deletions, effectively knocking out the function of the target gene. In the presence of a donor template, the cell may use homology-directed repair. This high-fidelity pathway allows for the insertion of new genetic sequences at the precise cut site. Off-target effects occur when Cas9 binds and cleaves sequences with partial mismatches. The kinetics of R-loop formation determine the probability of these unintended genomic modifications. Scientists have engineered high-fidelity Cas9 variants by neutralizing positive charges in the protein groove. This reduces the stability of off-target binding without affecting on-target efficiency. Mutating the D10 and H840 residues creates dead Cas9. This variant binds to DNA without cutting it, allowing researchers to visualize loci or modulate gene expression without permanent changes.
By fusing dead Cas9 to deaminase enzymes, researchers can perform base editing. This allows the direct conversion of one chemical base to another without inducing double-strand breaks. Prime editing utilizes a Cas9 nickase fused to reverse transcriptase. It writes new genetic information directly into the target site using an extended guide RNA template. In eukaryotic cells, Cas9 must navigate chromatin. Nucleosomes and heterochromatin structures can act as physical barriers, reducing the efficiency of the search and cleavage process. Naturally, CRISPR is a bacterial immune system. Bacteria capture fragments of viral DNA and store them as spacers within the CRISPR array to recognize future infections. For medical applications, the Cas9 system is delivered via viral vectors or lipid nanoparticles. These vehicles ensure the ribonucleoprotein complex reaches the nucleus of the target cells.
The thermodynamic stability of the RNA-DNA heteroduplex is significantly higher than that of the original DNA-DNA helix. This energy difference drives the displacement of the non-target strand. Cas9 nickases are engineered to cut only one strand. Using two nickases targeting opposite sides of a locus creates a staggered break, which significantly reduces the risk of random mutations. Other systems like Cas12 recognize different PAM sequences and produce staggered cuts with 5' overhangs. This diversity expands the range of addressable sites in the human genome. The duration of Cas9 binding influences the choice of repair pathway. Longer residence times may block certain repair factors, favoring error-prone pathways over template-directed repair. Within the cell, CRISPR complexes provide constant surveillance. This autonomous molecular machinery identifies and neutralizes foreign genetic elements through highly specific sequence matching.
After the task is complete, the Cas9 protein is eventually degraded by the proteasome. This ensures that the gene editing activity is transient, reducing the potential for long-term toxicity. The precision of CRISPR depends on the geometry of the active site. Even a sub-angstrom shift in the position of the magnesium ions can prevent the cleavage reaction from occurring. Engineering efforts continue to minimize the size of Cas proteins for easier delivery. Miniature Cas14 and Cas5 variant offers similar functionality with significantly smaller molecular footprints. The primary limitation of Cas9 is the requirement for a PAM sequence. Engineered variants with relaxed PAM requirements are currently being developed to enable editing at any genomic coordinate.
In summary, Cas9 cleaves DNA through a sequence of PAM recognition, R-loop expansion, conformational gating, and two-metal ion catalyzed hydrolysis. This mechanical process converts a genetic code into a chemical reaction. This ability to physically rewrite the code of life is transforming medical research. It provides a direct mechanism to correct pathogenic mutations at their source within the human genome. Unlike traditional drugs that target proteins, CRISPR acts directly on the source code. This fundamental shift in therapeutic strategy allows for the treatment of previously incurable genetic disorders. Every successful cleavage event is the result of billions of years of bacterial evolution refined by modern bioengineering. The result is the most precise tool for genome surgery ever discovered.
While the concept of genetic cutting is simple, the underlying biophysics involves a highly coordinated series of structural changes. Understanding these mechanics is vital for the continued development of genomic medicine. The chemical cleavage of DNA by Cas9 is a localized event with global consequences for biotechnology. It remains the primary mechanism for the direct and precise manipulation of biological information.